Hi, welcome to the Brain Gauge channel. I'm Mark Tomerdahl. I'm going to give you a brief introduction to the brain gauge right now. And the brain gauge is a laboratory grade research device that we developed in order to study brain function. So what does that mean? Well, it means that we crammed about $50,000 worth of laboratory equipment into a computer mouse-sized device, and basically it lost about 100 pounds over the 15 years that we spent developing it. And that device plugs into any computer or laptop with a USB port. And basically there's a software app that runs it, and it's fairly sophisticated. It's got over 3 million lines of code. So basically it's a laboratory that's been packaged very neatly into something that looks a little bit like a computer mouse. Now, why did we develop it? That's probably the most important question. And the reason there's several reasons, but we'll just go over a couple. One is, at the time we started developing it, there really were no decent ways to look at brain health or to objectively measure certain aspects of brain function. For example, one aspect of brain function is inhibition. And if you're not familiar with the brain is basically, its activity is made up of excitation and inhibition. So different parts of the brain turn on and different parts of the brain turn off. When it turns on, that's excitatory. When it turns off, that's inhibitory. And it's critical that the brain have a balance of excitation and inhibition. There are no ways to measure inhibition with any medical imaging method, but we can actually measure it with other methods, and this is one of them. So we can actually use this to measure one of them. We speculated on this back in 2004 and postulated that if we could develop this method, it would be extremely useful in research in many different neurological disorders, and that's proven out to be true. So let's go on to the conceptual idea behind this. And the main conceptual idea with the brain gauge is that if you stimulate two places on the finger, it'll activate two places in the brain that are side by side. And those two places in the brain will talk to each other. And the way they talk to each other will change what you feel. And basically it is a sensory perceptual task. And you can ask many questions about how things interact, how adjacent things organize, and you can learn a lot about things like plasticity and learning and memory by understanding the mechanisms that go into them. So what exactly does the brain gauge do? Well, if you look here in this picture, the brain gauge really doesn't do a whole lot other than vibrate the fingertips. But it vibrates them extremely precisely and accurately at specific amplitudes and frequencies and time intervals. So that's all been taken into account of, and that's based on years of research of what those timing sequences should be. And the brain gauge basically is a device that vibrates your fingers, and then when you deliver vibrations to the fingers, and what we do is we deliver a series of questions. Those questions are extremely easy. Which one is bigger? Which of the two stimuli are bigger? Which of the two came first? Which one lasted longer? So the questions really aren't that difficult conceptually, but the stimulus parameters may get more difficult, and they make things they're a little more complex, so that your answers to your questions tell us a lot about what's going on about specific mechanisms of brain function. So anyway, the results of those tests that we deliver are called cortical metrics, and they tell us a lot about dynamics of information processing. So after you collect all these cortical metrics, then what do you do with them? Well, this is an example of a case where we actually tracked someone's data post-concussion. So this person was tested on day 1, day 6, and day 14 after concussion. Day 0 is the day of concussion. And if you look over at this first plot, you'll notice that there's quite a varied group of bars. And intuitively, if someone glances at this, they usually say, red must be bad, green is good, short bars are bad, long bars are good. And if that's what you thought, you were correct. And you notice that from day 1 to day 6 to day 14, this person progressed until they actually were cleared to return to play. So besides just looking at the bar chart and the individual values, you can also look at the overall average. And if you look at the overall average, you see it right. You see it right here. Drawing a line over it. And you see that the overall history improves on this particular person from October up through late November of that particular year. And they got progressively better as time went on. So basically, this gives you an ability to track history of your performance on these tests. So, as I mentioned, we're able to use this in a lot of different neurological disorders, and the basic science and clinical research with brain gauge is going on in a lot of areas, like neurotrauma, such as concussion, which many people are familiar with just because of its prominence in the news, neurodevelopmental disorders, such as autism, neurodegenerative disorders, such as aging dementias, pain, pharmacological insult. So all these are areas that we're involved in, and we've got well over 60 publications that either utilize or validate these methods. And who uses the brain gauge? Well, right now, researchers in these areas, these different areas I just listed, use it. Biohackers use it, who want to see what effects of different supplements or nootropics or, their own hyperbaric chambers, whatever. Different people like to use this as their own experimental tool, because that's what it is. The brain gauge is an experimental tool that people use, either in their research or in their clinic or in their home. Okay, funding to develop this came from a lot of sources, and they basically kind of vetted us in order to get this. And most significant of all is the Office of Naval Research. They were the ones that really pushed us across the finish line in terms of getting this to a very stable technology readiness level. And that's pretty much it for this talk. And the future talks, I'm going to go over how to interpret the results and how to read different scores. Individual scores, I'm going to go a little bit more detail on. Also talk a little bit about history and talk about some of the other areas of research that we're involved in. I may take one talk just to talk about a specific paper, and also take requests. So if you have specific questions, please email me, and just email mark@corticalmetrics.com, or go to the website for more information and take a look at our blog or our publications. Thanks for listening.